class, let's go now on the first critical step, right? As far as you remember, we have the fixation of the preservation. Again, class, it is the process where we stabilize the physical as well as the chemical state of our tissues, of our cells, with the reagents that we use, so that there will be minimal distortion or decomposition of the organ. So, class, here, the aims of fixation is what I've told you earlier. It is to preserve the morphology and the chemical integrity of the cell in a life-like state. Please, class, lagyan ng star, lagyan ng heart, itong aims of fixation. Please take note of that. And the secondary one, it is to protect and harden the tissue, where it facilitates the easy cross-cutting and further handling of the sample. Okay? Again, first is morphology into a lifelike state. And then, secondary, it involves the protection and the hardening of the tissue. Okay? And the cross-cutting. So, class, here, the key reaction in fixation is we have the protein stabilization for cross-link formation. So this one, class, that I'm showing to you is the formaldehyde fixation, where it forms a covalent, right? Covalent bonds or covalent cross-links between the proteins that we have here and between proteins or between proteins and the DNA as well, based on this picture. So this preserves the 3D molecular arrangement of the tissue, and uh, it locks and the interactions, which eto class, it maintains the structural, I'm so sorry, I skipped the structural integrity of the sample. So what happened here, class, due to the chemicals that we have, there will be DNA modification, where it usually, again, it reacts with the DNA bases. Ano yung mga DNA bases natin, class? Do you remember? We have the adenine, we have the cytosine, we have the guanine. So, usually, class, etong tatlo, mayroon pa yung uracil, ha? So, usually, class, eto yung tatlo, it usually reacts like on the methylol products that we have here. Okay? So, the protein DNA cross-linking, it bridges the proteins to the DNA so that there will be a stabilization. There will be stability on their interactions. Ito plus quencher, there will be quenching. Okay, sorry. Why did I skip? Okay, umabot po tayo sa decalcification. Let me take a pause po muna, okay? Okay, class, balik tayo. Sorry. Ang ayaw ko umabot sa decalcification. So, what happened here, class, since there is quencher, there will be quenching. So, quenching agents, it neutralize the unbound formaldehyde to stop further reactions. Okay? So, eto po, reactions with quenching. Kikita nyo naman. Okay? Yeah. So, here, class, this is the living state. And this one, the PFA. Here, the aldehydes, reagents, and also here the next one, the methanol fixation. Okay, so here, class, on a living state, the proteins and the membrane here, they are surrounded by the dynamic water molecules. Ito pong color blue. So, here, class, the proteins can move laterally within the lipid bilayer and interact, they will interact freely with other molecules. So, our water here, class, again, they maintain the native hydrated state of that one. And ito naman, class, and here, class, we have the paraformaldehyde and glutaraldehyde, right? PFA and GA. So what happened here, class, is that the PFA, the paraformaldehyde and glutaraldehyde, they are aldehyde They are aldehyde-based fixative. So the penetration here, class, that this PFA, this PFA, ano ba yan, nabubul na? Okay, these PFA molecules will diffuse into the cells and tissues where they will be creating cross-link. Okay? Example class, like the PFA and the GA glutaraldehyde, they will react with the amino acids group, the example class, NH2 of the lysine residues and other nucleophilic sites on proteins. So this forms the bridges, okay, that we have here. And another one class, we have the methanol fixation. So unlike the PFA, the aldehyde-based fixatives, so the methanol class, they fixes the dehydrating cells, okay? Kaya ang alcohol. So it removes the water, which it causes the precipitation of proteins, okay, in place. So that one. So it's not really a true cross-linking compared to the aldehydes, but they still immobilizes the proteins here. So usually class these methanol, yeah, this methanol fixation, they are like often used in cytology and immunofluorescence for the permeabilization. Okay, I hope it is clear. Next is the effects of the improper environment. Of course, if there will be air exposure. Ano ba yan? Nakakahiya, ang daming mali. Okay, yeah. Exposure. It is when the tissue is exposed. Yan. The tissue exposed to the air. It leads to the shrinking and it dries out the tissue. That's why that there will be distortion of the cells that we have here. And it might evaporate and causing the cellular structures to collapse. And they will lose their normal shape, which it fails the function, the primary function of the, what we call this, the uh, fixatives, okay? Next is like the hypotonic solution. Of course, hypotonic, diba? Hypo, it will swell, right? So here, it has a lower concentration of solutes than the inside of cell. So this water, sorry, it move into the cell by what? By osmosis, which it leads to swelling. Okay? So that one, of course, in swelling, there will be lysis. Okay, cytolysis. And in hypertonic solution, okay, class, ito yung isotonic solution. So we are like, example, like an SS, like that. Anyway, in a hypertonic solution, our tissues here, our samples here, it will create a higher concentration of the solutes than the inside of the cell, where it will cause shrinkage. Okay, will shrivel. Okay, so here the shrinking of cell can disrupt the cellular processes and damage into the cell. Okay, so that is the effect of the improper environment that we have. And here we have different kinds of like additive fixatives here. Fixative becomes part of the tissue, forms cross-links, molecular complexes, formaldehyde, osmotic oxide, mercuric chloride. This is what we call additive. On the other hand, if it's about like alcohols, that they alter the tissue without being incorporated, stabilizes via dehydration, like earlier that I told you, we have the non-additive. Okay, I hope it's clear. So as we compare it like metaphorically, like the additive fixatives, they are like pouring glue on puzzle into pieces, like you can see here in this illustration. The fixative becomes part of the puzzle, locking each piece in place so that the picture stays intact. On the other hand, like this non-additive fixative is just like drying clothes under the sun. So the water is removed and the shape is set by shrinking and stiffening, but nothing here is added to the fabric. Okay, so there is just only dehydration. Okay, I hope it's clear. Plus, here like some factors that
effects like in fixation. Firstly is the hydrogen ion concentration, pH is why we have like pH meter, right? So the ideal one is around 6 to 8 pH, but the neutral one is around 7. The section thickness as well, class. Please take note that in electron microscopy, it must be 1 to 2 millimeter squared, and in microscopy, it's around 2 centimeters squared. And for this one, in usually class, around 4 millimeter thick or 0.4 point centimeter. Daming kulang. Okay, depending on the manufacturer. Totally depends. So larger tissues, they must be thinly and should be open, while the brain will be suspended in 10% MBF for 2 to 3 weeks. Okay, shall buttered formally. Then next we have the osmolality, which like I told you earlier, like the hypertonic, the hypertonic, the slightly hypertonic are 400 to 450 MOSM preferred, and sucrose is added. Please take note. The sugar sucrose is added to osmium tetroxide fixatives for the electron microscopy. What is added? Sucrose is added. What is added? To the osmium tetroxide fixatives for electron microscopy, it's the sucrose. Okay, the fixative concentration for formaldehyde usually it's around 10%. Butyraldehyde is 3. The immuno for electron this one immuno EM butyraldehyde at hold on 0.25 0.25 degrees percent. Okay, yeah, I hope it's clear. And we have to consider following. We have to consider our speed, our penetration. For the speed, immediate fixation, post removal prevents autolysis and putrefaction of our sample. A verification if bacteriologic and toxicologic tests are required, so it must be done first. And our penetration here formally penetrates tissue at around 1 millimeter per hour. Okay, and it varies depending on tissue type that we have. And they note class that our fixative volume must be around 20:1 for the ratio for the fixative to tissue. Pag tissue to fixative is 1 to 20. So it uses 10 to 25 times the tissue volume, max effective is 20. That's why 20:1. So osmium tetroxide only for 5 to 10 times due to high cost. One of the most expensive, osmium tetroxide, only 5 to 10 times due to high cost. Duration is, it varies by tissue structure. Ay, yung mga uterus natin class, intestines, they take longer to fix because they are fibrous. Fibrous organs. Okay, I hope it's clear. So here are the following characteristics of a good fixative. Pakibasa na lang po. Class, of course, it must be cheap, stable, and they're safe to handle. Okay, and they, like really preserve the morphology of it. Okay. Yeah, so fixation can be heated by the following. We could use the heat, vacuum, agitation, and microwave. And the ideal tissue size for fixation diameter not more than 3 centimeter. Thickness is not more than 4 millimeter. And the ideal fixation time is around 4 to 6 hours for the traditional. On the other hand, like for the others, like on the new references, like it's around 6 to 18 hours. So it totally depends. Okay? Yeah. So here, class, we have different types of fixation. We have the physical, chemical, simple, to like compound. Okay, like here for drying, physical. Here, just please take note of this one, please. Okay, so that will be the end of our fixation, but not all. Okay, so that will be only part one of our fixation.